Hi everyone, welcome back to my channel. This is lecture 3 of chapter Seeds, Structure and Germination, and today we are going to do topics like experiment on germination, types of germination, and study about what is seedling. So we'll study about seedling. So let's begin with our first topic, which is experiments on germination. In this topic, we are going to deal with 4 experiments. So let's discuss all the experiments one by one. So the first experiment is to prove that water is necessary for germination. We have to prove this thing. So suppose this is the beaker A which we have taken. This is beaker B which we have taken. Okay. Now in beaker A we have placed wet cotton. So here we have wet cotton. Okay. And in the second one, we have placed just normal dry cotton. Okay. Without any water. And then we have placed our any seed. Maybe pea seed or bean seed. Any seed you have placed here. So these are our seeds. Okay. These are the seeds. So over time what you will observe is that the seeds which are present in beaker A, they will germinate, while the seeds which are in beaker B will not germinate. Why? The reason being that in B water is absent, while in case of beaker A water is present. Hence this experiment proves that water is necessary for germination. So let's write this point. So here we can write in A germination take place. A germinates. So in B we can say does not germinate. Okay, and the experiment proves water is necessary for germination. Now let's see the second experiment. Second experiment is to prove that a suitable temperature is necessary for germination. To prove a suitable temperature is necessary for germination. Let's prove this. In this case also again we are taking 2 beakers. Okay. Let's draw with red. So this is beaker A. This is beaker B. Okay. And this time in both beaker A and beaker B we are keeping wet cotton. So water is available now in both the beaker. Okay. And we are placing what? We are placing our seeds. Bean seeds. Okay. Now what happens? Now we are keeping this beaker A at ordinary temperature. Okay. So A is kept at ordinary temperature. Whereas this B is kept in refrigerator. So B is kept in refrigerator. So here the temperature will be very low. Okay, and here the temperature will be normal. And then what happens over time, you will see that the seeds which are present in beaker A, they will germinate. because they are getting water and they are also getting a suitable temperature. While in case of B, although they are getting water, but here the temperature is very low. And in the previous lecture we studied that optimum temperature is necessary for germination, which is 25 degrees Celsius to 37 degrees Celsius. So here the seeds will fail to germinate. So again we can say that A germinates, means seeds in A germinates while seeds in B does not germinate. Does not germinate. So hence it is proof that a suitable temperature is necessary for germination. Okay. Now let's see the third and fourth experiment. So the third experiment is to prove that air oxygen is necessary for germination. To prove air means oxygen is necessary for germination. Let's see this experiment. Okay. In this we are going to take two conical flasks. Okay. It is sealed from top. In both we have taken wet cotton. Okay. And we have placed our seedlings. Okay. Now here we are taking a tube, an extra tube which is inserted inside this conical flask, and in conical flask A we have plain water. So in test tube of a conical flask we have normal plain water. Okay. Now while in test tube B we have taken a tube which is filled with pyrogallic acid. Okay. I will explain what is pyrogallic acid. It is filled with pyrogallic acid. So this pyrogallic acid, its function is to absorb whatever oxygen is present in that medium. So here whatever oxygen will be present in this conical flask, that oxygen will be absorbed by this pyrogallic acid, and therefore oxygen will be absent in B conical flask. Okay. So now what happens over time, you will see that seeds which are present in A, they will germinate because they are getting water. Okay. And they are getting oxygen also because of the reaction that is taking place here. Okay. And in this whatever oxygen was present, that oxygen is absorbed by pyrogallic acid. So here no oxygen is present, so the seeds in beaker B will fail to germinate. So let's write it down. In A, seeds germinate. Okay. While in case of B, seeds do not germinate. Okay, I hope it is clear. Now let's study about the fourth experiment, which is the 3 bean seed experiment. Okay. In this we are taking just one beaker. Okay. And on a scale which is kept inside it, we have tied 3 seeds, 3 bean seeds. So here one bean seed, here another, and here another. Okay. We have tied these seeds with this scale. Okay. and we have filled this beaker with water. We have half filled this beaker, okay, till here. Half of this middle seed is above and half is below water, okay. So this is the seed. Okay, this is the top seed, this is the middle seed, and this is the bottom seed. Okay, so what happens over time, you will see that in this experiment only the middle seed is able to germinate while the top and the bottom seed do not germinate. Reason being because the middle seed is getting both oxygen from the atmosphere and water which is placed in the beaker. Okay, the top seed does not germinate because it is not getting water, though it is getting oxygen. Okay, while the bottom seed, it is getting water but it is getting very less oxygen which is already present in this water. So B seed also fails to germinate. So let's write it down. Top seed, no germination. Why? Because it gets only oxygen, no water. Okay. Now in middle we see germination because it gets both water and oxygen. So it gets both water and oxygen. Now the bottom seed does not germinate, or it germinates later on due to whatever little oxygen that was already dissolved in the water. So B also does not germinate, so no germination, or germinates later you may say. Okay, because it gets water, and very little oxygen, very little oxygen. Okay, I hope the experiments are clear. Now let's see the types of germination. So basically there are three types of germination. The first is hypogeal, the second is epigeal, and the third is viviparous. Let's deal with all the three one by one. So first is hypogeal. Okay, the word hypo means below, and the word geo means earth. So what happens in hypogeal germination? The cotyledons, they remain under the ground, okay, and the epicotyle, it elongates and comes above the ground, and later on the radicle elongates. So let's first write it down. So cotyledons remain under the ground. Okay, second point is what? Epicotyle elongates. Let's draw a diagram and understand these 2 points. This is the ground, okay, and this is the cotyledons. From this cotyledons, the epicotyle, it comes out, forms the plumule which forms the shoot. So the epicotyle elongates, means the plumule elongates. Okay, and later on the radicals form root. So later roots will be formed. Okay, the cotyledons are under the ground, and the epicotyle elongates. This epicotyle only forms plumule which comes out of the ground and elongates further. Now let's see the examples that are given in your book. So the mnemonic is my PG. Okay. So M stands for maize grain. P stands for P. And G stands for gram. Okay. So this was all about hypogeal germination. Now let's study about epigeal germination. So the second is epigeal germination. Okay. The word epi means above and the word geal means earth. So in epigeal germination, the cotyledons are pushed above the ground and the hypocotyle elongates first. So let's write this point. Cotyledons pushed above the ground, okay, and the hypocotyle elongates first. So hypocotyle elongates. Okay, let's draw the diagram to understand it more nicely. This is the ground. Okay, so here the cotyledons have pushed themselves above the ground. So let's draw. So these are the two cotyledons. Okay, and here what? The hypocotyle elongates. This is the hypocotyle which has formed curved like structure and it has elongated. Okay, and later on the radicals forms the root first. Okay, then the plumule it elongates. So this is hypocotyle. Okay, cotyledons they are pushed above, they are seen above the ground and the hypocotyle elongates and forms a curved structure. So hypocotyle elongates first and after this the epicotyle which forms the plumule which further elongates later. So let's now see the examples of epigeal. The examples are BC. B is for bean and C is for castor. Okay. Now let's study the last type of germination which is viviparous. So the word viviparous means producing live young ones. Means producing live young ones. What happens in viviparous? The seed germinates inside the fruit which is still attached to the parent plant. Okay, earlier we studied that the seed was being separated from the fruit, okay, and later on in the soil it was forming a new plant. But in viviparous germination the seed is still present within the fruit and that fruit is still attached with the parent plant. So let's write. So in viviparous what happens? Seeds germinate inside the fruit which is still attached to the parent plant. Let's first understand it through the diagram. Okay, this is the stem of a plant. Okay, and here these are the leaves. Okay, and this is the fruit. This is fruit. Now, within the fruit, the seed must be present, okay? And that in that seed, the embryo, it elongates and tries to comes out of that seed. And from that embryo, the hypogeal elongates, the lower part of the embryo, which is known as hypocotyle, it elongates and the radicals forms the root. So in this we are going to draw, this is the radical which has come out of the embryo, okay? So this is the radical and this longer is the hypocotyle. Okay, this hypocotyle only forms the radical which later forms the root. So we can see in this that this radical and the hypocotyle, these are the part of the embryo, okay, and that are still attached to the fruit, okay, they have not separated from this fruit, and that fruit is still attached to the parent plant. This whole is the parent plant, okay. Now what you may do, you may later on plant this separately in the soil, and from that again a new plant will be formed. Now let's see the examples. So for the example of viviparous which are given in your book is of mangrove plants. Mangrove plants, they are found in the watery area. So in mangrove plants, two examples are given. One is rhizophora, okay, and the second is soneraceae. These are the two examples given, please learn them. So this was all about types of germination. We have covered hypogeal, epigeal and viviparous germination. Now let's study our last topic, which is seedlings. Seedling refers to a young plant. So germination in any plants ends with the formation of a seedling, which is a young plant. So we can write this: germination in any plant ends with the formation of a seedling. Okay, now let's see what is seedling. So seedling is the formation of a young plant from the seed. Okay, so let's write it down. Seedling is the formation of young plant from the seed, okay, which still depends on the food which is present in the seed, okay, which still depends on the seed for food to grow, okay. So seedling is the formation of young plant from the seed which still depends on the seed for food to grow. So what happens? The plant is still obtaining the food from the seed only. It has not become independent. So before becoming independent, whatever are the stages is known as seedling, okay, unless and until it has become fully independent to make its own food. So this was all about today's lecture. Thank you so much. See you in the next lecture.